For the males, we have three slides for you. You have the testis, you have the epididymis, and you have the penis. And so this slide is going to be familiar from lecture. This is the testis. And what we can see here within the testis are two seminiferous tubules. And these seminiferous tubules are those tubules where sperm develop. So if we consider this one here on the left, we have this outer area. This is sort of the basal layer or the germinal layer. And along this outer area, this is where we have the least mature. These are the kind of the earliest of the developing spermatocytes. As they divide and go through meiosis and become more mature, they start to acquire a more typical sperm-like appearance. And so by the time you get to the lumen, you have cells that are referred to as spermatozoa. And these, although they resemble kind of the mature sperm, are still immature in terms of their ability to fertilize an egg. These cells are incapable of swimming at this point. They're going to be moving due to the pressure that's being built up as fluid is added to this lumen. So again, the cells along the kind of the outer edge here, these are the immature. And as you move towards the lumen, you're seeing more and more maturity. And even in the center, you're seeing these spermatozoa that are still incapable of swimming and fertilizing an egg, but they're much more mature than the original ones around the outer edge. The other cells that make up these seminiferous tubules are the sustentacular cells. And these are the cells that I mentioned in lecture are actually quite large, with then very, very large prominent nuclei with then nucleoli inside them. So here we can see this is a nucleus. And then down here we can see a couple other nuclei of the much larger sustentacular cells. And these are the cells that create the blood-testis barrier, the tight junction between them, as well as then is going to be producing nourishing fluid into the lumen that will help nourish and support these developing spermatozoa. So the sustentacular cells we can see by these kind of large columnar nuclei with very prominent nucleoli, whereas all the other cells in here, these are just developing spermatocytes at various stages of development. Then the third cell type that you're expected to be able to identify within the testis slide would be the interstitial cells. And so the space here outside or between the tubules, this is where you're going to find the interstitial cells. And these are these cells here that have the nice kind of granular cytoplasm with very prominent nuclei. These interstitial cells are the ones that produce testosterone. They're producing the sex hormones. So again, we have blood vessels out here. This is going to be the vascular tissue around the seminiferous tubules, and it's within here that you'll find these very kind of prominent cells with very nice large nuclei. These are the interstitial cells. As we move on to the second slide, we're into the epididymis. And the thing to notice here is it's also just a big collection of tubes, and more specifically, it's one really long tube. So the duct of the epididymis, or the ductus epididymis, is a single tube that's going to be present in each testis, actually just outside the testis. And it's in here that the spermatozoa are going to mature. So they may spend a few weeks within the epididymis, and more specifically the duct of the epididymis, and this is where they're going to mature. If we look closely at one of these ducts, we can see some features that should hopefully distinguish it from the seminiferous tubules. One thing you'll notice a lot of spermatozoa present within the lumen. So these again are moving through this long epididymis, again acquiring the characteristics that allow these sperm to swim and fertilize an egg. The other thing you'll notice is the wall of this is going to be a pseudostratified columnar ET, and they're going to have these long microvilli or the stereocilia. And these are not here to move the fluid through. They're actually here to add surface area for absorption of the luminal fluid as well as secretion of nutrients to help nourish and support these developing sperm. So you'll notice the wall is very different when you compare this tube, the duct of the epididymis, with then the seminiferous tubules that you'll see within the testis itself. Both of them should have spermatozoa or developing sperm within the lumen, but here the wall is going to be appearing to be this pseudostratified as opposed to a layer composed of many cells of developing spermatocytes. The female reproductive system, there are only two slides, although they tend to be a little bit more complex. The first one here is the ovary, and what you're being asked to identify here are the various stages of ovarian follicle development. And so within this cross-section, out here, this is going to be the outer surface of the ovary, and it's out here that you're going to find the germinal epithelial layer. This is a simple cuboidal layer, so we would need higher magnification to really see it. As we look within the ovary itself, you'll see in this case dozens, maybe hundreds of different follicles, all at different stages of development. And so the four that you're looking for, there's primordial follicles. These tend to be the smallest, and they often are out here on the outer edge. Then we have primary follicles. These right here would represent some of the primary follicles. And then we're going to see some secondary follicles. These two right here are nice examples of secondary follicles. You can see this little space that's forming in there. And then finally, we have these mature follicles where that space is much, much, much more prominent than in the secondary. So if we look at some of these in a little more detail, first, if we look around the outer edge, we can see a bunch of these very small follicles here. And a lot of them, if you maybe can see over here, there's really only a single layer of cells that surrounds the oocyte. So these are all the primordial follicles. And these are the ones that a female would have at birth, and maybe a million or so of these at birth. These are all arrested in a very, very early stage of meiosis. So there's this single oocyte here, typically surrounded by a single layer of cells around it. And so these are all these small follicles here. As we look more closely at some of the larger follicles, we should start to notice then some layering going on. And so if we take a look at this one, for example, everything within this blue line right here, this blue line is referred to as the zona pellucida. And this is this extracellular space, this kind of gelatinous material that surrounds and protects the oocyte. So everything inside that blue line, this is the oocyte. And then we have the zona pellucida. And if it is a primary follicle, which this one is, there will be several layers of granulosa cells that surround it. And so this ring of cells here, there's probably 2 or 3 cell layers thick. These are the granulosa cells. And so these that have the oocyte with the zona pellucida and then a few layers of cells surrounding it, these are all primary primary follicles. And so these all right here, these would all represent these primary follicles. For them to become secondary follicles, you're going to want to look for this space. And the space is referred to as the antrum. So here you can see a little space here. This one over here we would also call a secondary follicle because of the presence of this antrum. And as this antrum is forming, we will now start to give these cell layers a different name. So we have the oocyte in the middle, the blue zona pellucida, and now these granulosa cells that are directly outside the zona pellucida, this is going to be referred to as the corona radiata. So the corona radiata refers to this layer of cells directly outside the zona pellucida. And then outside that is where the antrum is forming. So this antrum is going to then exist outside the corona radiata. And then outside the antrum is then another layer or layers of granulosa cells, and these are called the granulosa cells. And so the granulosa cells in the middle, those now are called the corona radiata, whereas outside we continue to call these the granulosa cells. And then you'll also notice there's this layer of kind of dense tissue, almost looks like a nice solid band that wraps around it. These are going to be called the thecal cells. And so the outer layer here that surrounds this entire follicle, these are the thecal cells. And so for a secondary follicle, you should see the oocyte, the zona pellucida, corona radiata, antrum, granulosa cells, and then this outer area here. These are the thecal cells. And when we go from secondary to mature follicle, the big difference is just the size of the antrum. So here we can still see the same layers: oocyte, zona pellucida, corona radiata, antrum, granulosa cells, and then the thecal layer outside it. And so the only difference here is just the relative size. If you could fit maybe 3 or 4 of these oocytes in here, then we'll call that a mature oocyte. Sorry, again, I keep saying that, mature follicle. The last follicle we have, this is the corpus luteum. This is after ovulation. So those mature follicles, those are the ones that are ready for ovulation. That oocytes can be released. And after that, these essentially empty follicles, the antrum, fills in with some granulosa cells and blood and other tissue. And when that happens, they're referred to as the corpus luteum. These tend to be these large, almost kind of, they resemble in a sense these mucus glands. They have these large cells that almost look kind of frothy in a sense. These are the corpus luteum, and these are these follicles that follow ovulation, and these are going to be very important at secreting some of the sex hormones that will then influence the uterine cycle that we'll talk about in lecture. And so the last one here, the corpus luteum. And then the final slide we have is going to be of the uterus, and there are only a few terms related to the uterus. There's the 2 layers that are relevant. The inner, I guess if you think about it, here's the lumen of the uterus. And so this would be the center of the uterus. This mucosal layer, this is going to be referred to as the myometrium. Sorry, I take that back. It's the endometrium. Get a little ahead of myself. So the endometrium is this layer here that's going to be the mucosa. There's really no submucosa in the case of the uterus, and so the mucosa layer is referred to as the endometrium. And you'll notice in here that there are a lot of glands. So these are these uterine glands that are going to be present throughout the endometrium. And we're asking you to distinguish between these two regions. There's the stratum functionalis and the stratum basalis. And really, it's hard to see a distinct line between these. But if you were to have a pointer asking you about this kind of basement layer, so here, right here, this is the transition out of the endometrium into the smooth muscle layer or the myometrium. And so the part of the endometrium next to the myometrium, where this arrow is, this is going to be referred to as the stratum basalis. This tends to stay around. This is not the part that's going to be lost during menstruation. Whereas the functionalis, this is the part of the endometrium that's near the lumen. This is the part that kind of grows and shrinks over time depending on hormones, and this is the part that will be shed during the menstrual cycle. And so we have the endometrium, which is the mucosa, and more specifically, there's the layer, the functionalis. That's the part that kind of changes in shape due to menstruation. And then there's the basal or the basalis layer, and this tends to be retained. And then down below, we have the myometrium. This is the smooth muscle layer of the uterus. Here's just a close-up of that. We can see the uterine glands. These uterine glands then open up to the lumen, and this would all kind of represent the mucosa layer with the lamina propria. This may resemble actually some lymphatic follicle that's existing within the uterus. And this last slide, this is another one of the uterus, and we can imagine there would have been the lumen up here, and so I've cut the slide off, but the lumen would be out on the top here. So this is the endometrium from here to here, and then this becomes then the smooth layer, muscle layer, the myometrium. And what this one is showing is that depending on the different phases of the uterine cycle, we will have different appearance of these glands. So here we can see these glands are taking on a much more spiral appearance, and that's due to, again, the phase of the uterine cycle.